Hi, I'm Walt Hitch. I'm the director of the University of Tennessee's Plateau Research and Education Center here in Crossville, Tennessee. And today we're focusing on some of our research highlights. And today we have with us Samantha Roberts, and she is a sophomore at the University of Tennessee Animal Science Department in Knoxville. And many times we have master's and PhD students, but Samantha is a sophomore, and I'm going to let her introduce herself.
I'm Samantha Roberts. I'm a sophomore at the University of Tennessee, Knoxville. I'm from Athens, Tennessee, and I'm working in Dr. Moore's lab.
Can you tell us a little bit about your project?
My project aims to help identify replacement heifers that have high and low fertility, which is a key component for beef cattle production.
Can you tell us why that's a key component for beef cattle production?
Heifers that breed late or do not breed at all have a huge financial impact on farmers. Heifers that breed late, they have lighter calves at weaning, and they will most likely have a harder time breeding back for that second calf. This way, if they do this, they almost can miss a second year's worth of calf production.
I would agree, Samantha, that getting heifers bred, and even bred that second time, may be one of the biggest challenges to beef producers. But what about those that, not just the ones that get bred late, but the ones that don't get bred at all?
The ones that don't get bred at all, you put so much time and effort into feeding them and getting them the right weight and be able to make sure they have mineral out to make sure they will be able to get bred. But usually when they're diagnosed open, they're sold.
Okay, so once again, just a huge impact to get those animals bred and bred early as a replacement heifer.
Yes, sir.
So with this project, who else is involved in this project?
We're working with scientists from all across the country, from Tennessee to Montana to Virginia, to identify genes in heifers' blood cells that is connected to heifers of high and low fertility.
So have there been any previous experiments done in this area?
Yes, we have previously collected blood at AI and have detected a difference in gene expression between heifers that caught at AI and those that did not breed during that breeding season.
So would you please tell us, what is gene expression?
With gene expression, we are looking at how much RNA for each gene is in a sample of the heifer's blood. A heifer's DNA has multiple genes that have varied products and functions for growth, bodily functions, and even reproduction. The RNA is a messenger for each gene-specific product or function. If there's a lot of RNA for a specific gene, that means the gene's function is really active in the heifer.
Okay, that makes sense. Why are you interested in comparing the gene expression between heifers of high and low fertility?
The different amounts of RNA in heifers that catch at AI and those that do not help us understand what biological functions may be different in these two groups of heifers. We also believe there could be a potential to one day develop a fertility test related to the amounts of RNA in the blood that could be connected to infertility.
Okay, so how does the Plateau AgResearch Center help you with your research?
Complete this project, we need to include more heifers. We would also like to see if this test could be performed at a younger age, like at weaning. We are collecting blood from PREC and multiple RECs in Tennessee at weaning age, before the breeding season begins, and even at the time of breeding. The REC employees play a huge role in this project. They help us by getting the heifers through the chute, giving us background information on the heifers through their records, and by even breeding the heifers.
Okay, so you mentioned collecting blood. So what happens to the blood after you collect it?
After we collect the blood at each of the stages of collection, we take it back to the lab to be processed. Then we will compare the different gene expressions between the heifers that caught at AI and those that did not get pregnant during that breeding season, to see if the gene expression exists.
So what is the end result that you are aiming for in this project?
We hope that our group will be able to use this information and one day develop reliable means for producers to detect and sell infertile heifers at an earlier time.
Okay, this is all very interesting, and I think it's just fantastic that you're doing this even as an undergraduate student, and we're glad to support you in doing that, and we're glad to have you as part of our research highlight focus today, and wish you the best, and in your career and in your degree. So.
Thank you.